Saporta. I'm a neurologist at the University of Miami. I've been working in CMT both clinically and in research for probably almost all my career. And I'm here to chair the session on type 2 CMT. And I took my talk to really focus on kind of laying the ground for what the other speakers will speak about. So really explaining what CMT is, how we classify CMT. Because at the end of the day, I feel there's a lot of confusion when we talk about type 1 CMT, type 2 CMT, autosomal dominant, autosomal recessive genes. So we're going to try to create this kind of common language so that we can all talk the same language, essentially. So I'm going to give this kind of initial presentation. Then Dr. Bipasha Mukherjee-Clevin from Johns Hopkins will talk about their work in a specific type 2 form of CMT. And then Thomas Cronenberg from NMD Pharma will talk about their work, which it kind of spreads more broadly across CMT types, but have a particular interest in CMT type 2. All right, so that's kind of my talk. So every time we talk about CMT, we are basically speaking about a peripheral neuropathy. And peripheral neuropathy is a fancy way to say nerve damage. And when we're talking about nerves, we're talking about this part of our nervous system that connects our brain and our spinal cord to our muscles and our skin. When we talk about Alzheimer's, Parkinson's, stroke, we're really focusing on the brain. CMT, in the majority of cases, do not affect the brain function, right? So we're really talking about peripheral nerves, and I'm going to give a little bit of a zoom image here. And peripheral nerves are particular in the sense that they have to connect our central core of our body to our extremities. So they have to deal with very long lengths. If you think about our leg nerves, they are going from our lower back. It's kind of hard to see the pointer on the screen, but our lower back over here, all the way down to our foot muscles. And that's over 1 meter long. So there's a lot of metabolic demand, meaning energy demand, to make this system work. And we think about peripheral nerves, we can compare them to kind of our electrical grid, right? So we have a place where energy is being produced, and that energy has to reach our homes, usually hundreds of kilometers or miles away from that energy source. So our peripheral nerves work like this kind of electrical grid, and they are, of course, susceptible to the same issues of if there's a storm and it knocks down some of those wires, of course, the energy will not go to where it's supposed to. The same thing happens when our nerves are injured. And peripheral neuropathy is a very general term. CMT is one form of peripheral neuropathy, but there are probably hundreds of different types of neuropathy. Everybody probably heard about diabetic neuropathy, so that nerve damage that's caused by diabetes. CMT is a different type, but every time we're seeing a patient for the first time, especially if they don't have a family history, we have to figure out, is this actually a CMT case, or could this be another form of peripheral neuropathy? So everything that we kind of be talking about is trying to really make this kind of diagnostic process of seeing somebody with signs of a peripheral neuropathy, which you guys are very familiar with, the foot drop, numbness in their feet, weakness in their hands. Those are the clinical symptoms, but then what's causing the nerve damage or the neuropathy is what's going to define if a patient has CMT or not, okay? So just like we use the electrical grid analogy to kind of explain the peripheral nervous system, we usually use the electrical wire analogy to explain how peripheral nerves are organized. So in wires, you have kind of the inner part, which is metal, and it's where kind of the electricity is going through. But it's very important to have some form of insulation so that the energy doesn't dissipate away and kind of it's lost during that process of transmission. The same thing happens with our nerves. So we need something to isolate our inner part of our nerves, which we call axons, and which will allow for transmission to happen in a more efficient way, but also will help support that inner part, that axon, so it doesn't suffer issues of how it functions. So in this drawing here, you're seeing the axon in yellow, which kind of is an analog to the wire part of the inner part of the electrical wire. And then the insulation, which is here is like this colorful rubber parts, in the peripheral nerve is the myelin. So the myelin sheath is produced by Schwann cells. So we're going to always be using Schwann cell and myelin kind of interchangeably to just convey the idea that the problem is not in the inner part of the axon primarily, but actually on the insulation of the nerve, okay? And that will be a very important distinction that we're going to make in the next few slides. So I use this kind of to ask you guys what you guys are seeing on this kind of figure. So what are those animals there? Birds, right? But they're very different birds. So why do you think those are birds? So what are you using to define them as birds? What are the features that tells you that those are birds? They have feathers, they have beaks, right? So they have common things that will make us group them together. But if you look at them individually, they're very different from each other. And that's kind of how CMT is as well. So we use certain features to name something CMT, but of course we're clumping together a bunch of different conditions that are very similar in certain aspects, but they're very different in others. So for us to call a patient, or diagnose a patient with CMT, we have to determine that they have nerve damage or neuropathy, and we have to somehow prove that they have a genetic problem or a DNA problem. And that DNA problem is the cause of the nerve damage, right? But just like here we are seeing at least 20 different birds, we have now over 100 different types of CMT based on where the genetic problem is, right? So there are those two ways to classify CMT. One is based on what pattern of nerve damage we find in the patient, and then if we are able to determine where the genetic problem is, okay? In regards to nerve damage or neuropathy, our main distinction will be between the two structures I mentioned. Is this a problem of the axon, the inner part of the nerve, or is this a problem in the insulation or the myelin? If it is a problem of myelin or Schwann cells, we call it CMT type 1. And this is not the type 1 session, so if you guys have type 1, you should go to the other room. But if the problem is in the inner part of the axon, we call it type 2. And this can be a little further confusing because eventually type 1 CMTs will affect the axon because that myelin needs to be there to support the axon. So type 1 patients will have axonal issues as well, but primarily the problem kind of arose from a problem with the myelin, where in axon CMT or type 2 CMT, the myelin is kind of functional and normal. The axon is where the problem is. Now, if we do a genetic testing and find a specific mutation, and when we talk about mutations, we're talking about a change in the sequence of our DNA kind of recipe, then we can classify the CMT very, very specifically. This is type 2A, this is type 2F, this is type 2E, based on where the mutation is, in which gene it is. But sometimes the only thing we can determine is that there's a family history. We can tell by looking at the family that grandma had it, then father had it, then the patient had it, and their siblings and children. So what we try to do then is to classify them in what type of inheritance this looks like. And there are two major types. There are others, of course, and I didn't include it here to not overcomplicate things, but the two major distinctions is between autosomal dominant and autosomal recessive inheritance. When you talk about autosomal dominant, we're talking about a situation where a single change in the DNA is enough to cause a problem. And recessive, you have to have changes on both copies. And just kind of going back a little bit, it's important to remember that we are all, in many different ways, the product of our parents. And from a genetic perspective, that's very, very true because we receive half of our genetic code from our father and half of our genetic code from our mother. And that the kind of obvious conclusion about that is that most of our genes are doubled. So we have two copies of every gene, one that came from our father and one from our mother. In autosomal dominant, a single change in one of those copies is enough to cause disease. So here we have dad who has a disease, has CMT. He has this mutation here in the light color. And then mom is not affected, so she has two normal dark copies of the gene. Then when they have their kids, every time dad would pass that mutation to one of their children, that children will have a risk of developing the disease. If he passes his other normal copy, then they won't develop disease. And that's why we say when we see that there's an autosomal dominant condition, there's a 50% chance of a kid to develop disease because there's a 50% chance of inheriting that mutation, okay? For recessive, it's different because you have to have affect two abnormal or two mutated genes to actually develop disease. And that's kind of shown on the last children here, where she inherited the mutation, which is here. It's the boy here. The mutation is in blue. So he inherited one mutation from the father, one mutation from the mother, and then he has a disease. But the parents themselves don't have it because they only have a single mutation, each one of them, okay? For most type 2s, which we're going to be talking here today, we're talking about autosomal dominant conditions, but there are exceptions. And the most famous one recently is SORD, because SORD, when we look at the where in the nerve the problem is, it's classified as an axonal type or type 2, but it is an example of a recessive condition where parents will not be affected, but then their children will have a 1 in 4 risk of developing the neuropathy, okay? And the last thing I want to talk about is how a mutation in the DNA actually becomes a problem, right? So this is kind of how our genetic code works. So we have a primary recipe, the DNA, which is where our genes are. The DNA has to be transcribed into an intermediate form called the RNA, and then the RNA becomes a protein, and the protein is actually the building block that makes our body. Now, that protein will have different biological effects, and if a mutation happens and that biological effect is kind of messed up in a way, then you can have, especially in CMT, long-term consequences. And that can be loss of the axons that we talked about, loss of muscle mass or muscle atrophy, inflammation. There are different things that will happen kind of down the line and could potentially be therapeutical targets, as we're going to see in the next talk. But they're not the primary problem, meaning they're not the genetic problem, right? So every time we're talking about a genetic disease, we're really talking about a mutation in the DNA that becomes a mutated RNA, that becomes a mutated protein. That mutated protein either lost its normal function or gained a toxic extra function, and then that will cause the nerve damage and eventually all the downstream or the long-term consequences related to the neuropathy. And I'm showing this here because this is kind of how we approach treatment for CMT, right? So those are all points where we can intervene to try to treat CMT. So you guys probably heard about CRISPR, which is genome editing or a way to fix the mutation in the DNA itself. You also probably heard about gene replacement therapy, which is basically putting a piece of DNA back into the body when that gene is missing its function. So those are kind of DNA level treatments. You can also target the mutated RNA and either prevent it from being produced or trying to somehow correct it in a way that makes up a normal protein back into the body. And those are we try to accomplish that using antisense oligonucleotides or siRNA. So there are different tools to change how this RNA is expressed, either preventing it from being expressed, which is kind of the way we try to treat CMT1A, or modulate its expression somehow. And then you have the more traditional types of treatments, what we think about when we think about drugs or medications, which we call small molecules. And small molecules can have a very kind of wild range of applications. They could be targeting DNA and how DNA becomes RNA, how RNA becomes protein, or then target one of the biological effects or even the long-term consequences of that damage that happens in the peripheral nerve. And this is kind of why you see this gigantic list of projects that are being supported by the CMTA. Here, there's this first distinction between type 1 and type 2 and other forms of CMT because they are different conditions, both from a genetic perspective but also a biological perspective. But you're going to see different designations here based on the fact whether this approach is a small molecule, it's a gene therapy, it's CRISPR, it's an antisense oligonucleotide. So just to show you that we are trying everything, like every different approach possible. And the next talks will be focusing more on the small molecule kind of side of things, but we do have efforts going on for all the different types, and we'll be happy to talk about them on the Q&A part of the talk. All right, let's see how I'm doing. Okay. So we're going to move now to our first talk on projects involving CMT type 2. Dr. Abhipasha Mukherjee-Plavin from Johns Hopkins University will talk about their work on CMT to see CMT. Okay.
Good morning. Thanks so much for your attention, everyone. And thank you so much for giving us a nice introduction to CMT and axonal CMTs in particular, because I think you set the stage really nicely for my talk, Dr. Saporta. So today I'd like to share with you a story that's specific to Johns Hopkins University for one specific form of axonal CMT, and that's called CMT2C. And so I'm going to share with you the story of the TRPV4 Natural History Study, which is a study that is ongoing at Johns Hopkins University. But before I dive too much into the details of the study, I want to make sure that I acknowledge the folks who have done the work that I'm presenting today. So much of this work was led by Dr. Brett McRae, who was at Johns Hopkins University up until recently, and he's now at the University of Michigan, where he is starting his own CMT clinic. The infrastructure for this study was laid down by Maya Hoke, who is now a medical student at the University of Buffalo, and we're very proud of her. And all of the work that I'm presenting to you today was done under the leadership of Dr. Charlotte Sumner, who's on the CMT Association STAR Advisory Board, and she's the world's leading researcher on TRPV4-related neuromuscular disease, or CMT2C. So CMT2C, or TRPV4 neuromuscular disease, as we like to call it more broadly, is an unusual form of CMT. So basically, these patients, what they have in common is they all have a mutation in the TRPV4 gene, which then results in a mutated TRPV4 protein. And that mutated TRPV4 protein can then go on to cause a number of different symptoms. And what these patients all have in common is that they have an autosomal dominant condition, meaning they inherited an abnormal copy of the gene from one of their parents, and there's a 50% chance that they will pass it on to one of their children, as Dr. Saporta said. And all of these patients also have muscle weakness, and they may or may not have sensory loss. The pattern of their muscle weakness can be pretty variable. So there are patients that we label as having CMT2C because their muscle weakness is the most severe in their feet and in their hands. So their longest nerves are affected, as Dr. Saporta said, is common in CMT. However, there are other patients with TRPV4 mutations who actually have pretty strong hands and feet, but have significant weakness around their shoulders and around their hips. And we label those patients as having scapuloperoneal SMA. And then there are other patients who are basically born with weakness, and we label those patients as having congenital distal SMA. What we're finding, however, is that most patients with TRPV4 mutations don't neatly fall into one of these three categories, and rather patients with TRPV4 mutations sort of exist on a spectrum. And so we like to also refer to this condition as TRPV4 neuromuscular disease. So in addition to having variable patterns of weakness, the patients with CMT2C or TRPV4 neuromuscular disease can also have some other distinct features that are not common in other forms of CMT. So they can have prominent scapular winging, which means that their shoulder blades can stick out quite prominently because of significant weakness and atrophy of their shoulder muscles. They can also have problems with eye movement abnormalities. Sorry, let me go back. Oh, all right. Well, they can also have problems with eye movements. They can also have problems with vocal cord function. They can also have problems with hearing, and they can also have problems with neck muscle weakness. And this slide doesn't show it, but they can also have problems with diaphragm weakness and bone development. Here, I'm going to show you an example of a little 2-year-old girl with TRPV4-related neuromuscular disease who's quite severely affected. So she has significant weakness in her arms and legs, but she also has bilateral vocal cord paralysis, which means that her vocal folds, which typically should be moving in and out as we breathe and as we speak, in her case, her vocal folds are paralyzed because of this condition, and so she can't breathe well. Let me see if I can play this video. Are you all able to help me play the embedded video in the slide? There we are. So this is us just listening to her breathe. She has something called severe inspiratory stridor. Because her vocal cords are paralyzed, when she takes a deep breath in, her vocal cords aren't able to move apart to create room for air to come in. And so when she takes a deep breath in, the air that she's trying to breathe in is meeting a lot of resistance from these paralyzed vocal folds. And so you can see here her vocal folds aren't moving well. And then earlier you could hear that really sharp stridorous sound. So you can get a sense that this is really, really disabling and uncomfortable condition for this little 2-year-old girl to be in. Dr. McRae sent a questionnaire out to 37 patients with TRPV4 neuromuscular disease, asking them what their most bothersome symptom was. And what we found in the survey is that many patients with CMT2C or TRPV4 neuromuscular disease are really bothered by the significant weakness that they have around their hips and thighs, or their upper arms and shoulders, or breathing problems. So you can see in bright green is the category of patients that have significant breathing problems. And so this information tells us that these patients have some things in common with other forms of CMT, but then they also have some distinct features as well. I think the really exciting thing about TRPV4 neuromuscular disease is that it's a form of CMT that's actually potentially treatable. So Actio Biosciences in San Diego, California, has developed a small molecule TRPV4 inhibitor to treat TRPV4 neuromuscular disease. They think that this inhibitor targets all of the disease-causing TRPV4 mutations with high potency. And what they're trying to develop is basically a pill that's taken by mouth, hopefully once a day, to try and treat TRPV4-related neuromuscular disease. They're currently testing their potential medicine in TRPV4 mouse models. And if everything looks good, we at Johns Hopkins are hoping to partner with Actio Biosciences to test this compound in a therapeutic clinical trial to see if it can help improve symptoms in patients with TRPV4 neuromuscular disease. However, with where things stand right now as far as our knowledge of TRPV4 neuromuscular disease, it would be really hard to confidently design a therapeutic clinical trial for this type of CMT. And some of the challenges in trying to confidently design a therapeutic clinical trial for this type of CMT are the fact that these patients have a very rare and heterogeneous disease. We lack an understanding of the natural history of CMT2C, meaning we lack an understanding of how this form of CMT progresses over time. We also lack an understanding of the full phenotypic spectrum of this disease. And the field currently lacks biomarkers of disease severity and progression to appropriately design a clinical trial with confidence. And so to try and address these barriers, at Johns Hopkins, we're currently conducting the TRPV4 natural history study. So as part of this natural history study that's currently unique to Johns Hopkins, we're inviting patients with established TRPV4 neuromuscular disease who might live anywhere in the United States to fly to Johns Hopkins to participate in this 6-year natural history study. We're asking these patients to come to us every year for 6 years for an intense 2-day battery of testing. And our goal enrollment is 60 patients. And so far we've enrolled 12 patients. And if you're interested, recruitment information about this study is actually listed on the CMTA's website as a patients as partners in research opportunity. During the 2 days that these patients come to Johns Hopkins, they undergo a battery of these evaluations. So they undergo a clinical neuromuscular examination. They undergo vocal cord assessments by one of our ear, nose, and throat colleagues, a voice assessment by one of our speech-language pathology colleagues, an EMG nerve conduction study that's done by me, so I meet every patient who comes in for this study. They undergo breathing tests, study questionnaires about symptoms. They undergo 2 types of bone scans to look for bony abnormalities, a special research protocol MRI of the brain to look for blood-brain barrier dysfunction, because we think that that's important in this particular form of CMT, as well as other physical exam assessments and blood and urine testing. And it's a lot that we're asking of these patients, but I think that this work is important and impactful. We plan to use the information that we get from this TRPV4 natural history study to ultimately design the therapeutic clinical interventional trial for TRPV4 neuromuscular disease in collaboration with Actio Biosciences. And I think that this work is really important and really exciting to me personally for 2 different reasons. One is that I think that the work that Actio is doing represents potentially one of the first disease-modifying therapies for a form of CMT. And then second of all, I think that there are going to be lessons learned from the way we do this natural history study and the way we ultimately design the therapeutic interventional clinical trial that will be informative for the way we do clinical interventional trials for other subtypes of CMT. So I think this is a really important work, regardless of whether or not each of us is affected by CMT2C specifically. So with that, I'd like to thank you all for your attention. We're, of course, actively enrolling. So if you or anyone you know has TRPV4 neuromuscular disease and you're interested in contributing, feel free to reach out to me. The email address for Sam, our study coordinator on this study, is also there as well. And of course, none of this work would be possible without the patients and families who are affected and so invested in helping contribute to finding a cure. Thank you.
Okay, so next we're going to hear from Thomas Grönbeck from NMD Pharma about the development of a potential treatment for CMT. Thomas.
Thank you so much. And thank you so much for the opportunity to address you all here at this very exciting meeting. I think it's been wonderful so far. We have learned a lot, and we continue to learn from meetings like this. My talk will be about the clinical development of CLC1 inhibitors for the treatment of Charcot-Marie-Tooth disease. My name is Thomas Grönbeck, and I work as a clinical scientist for NMD Pharma. So NMD Pharma is a clinical stage biotech company. We are based in Aarhus, Denmark, which is the 2nd largest city in Denmark, about 2.5-hour drive from Copenhagen. The company is founded based on more than 20 years of research in ion channel biophysics at Aarhus University. Here, Thomas Holm Pedersen, our current CEO, was and actually still is an associate professor at the university, and it is based on his research that the company is eventually founded. The company was founded in 2015, and today we are almost 50 employees across Denmark, Spain, UK, France, and the US. We are funded by venture capital funds, and our main investors include Roche, Lundbeck, Novo Nordisk, Inkev, and Jaito. So this slide here is showing you our growing pipeline of different treatments for different neuromuscular disorders. And I would like to draw your attention to the top 3 lines on this pipeline chart, which is showing you where we are in the development with a drug called NMD670 for 3 different neuromuscular diseases. And those diseases are myasthenia gravis, it's spinal muscular atrophy, and of course, most importantly to this meeting here, Charcot-Marie-Tooth disease type 1 and 2. We also have other treatments in the pipeline, other CLC1 inhibitors that have a slightly different profile compared to NMD670, and then completely other drugs that are targeting different pathways. But NMD670 is a CLC1 chloride channel inhibitor. It is formulated as an oral tablet. It's supposed to be taken 2 times a day, approximately 6 to 8 hours apart. And how it works is that it enhances skeletal muscle activation when the communication between nerves and muscle isn't working as it should. And we'll get into more details with that in a moment. We hope that this drug will increase muscle strength and power and endurance when these are things that are lacking. So this slide here is showing you in a little bit more detail the mechanism of action of this drug. You see a nerve signal that is transmitted from the brain via the nerves and to the muscles. And at the very end of the nerves, you have the connection between the nerve and the muscles. It's a highly specialized synapse called the neuromuscular junctions. And this is where nerve signals are transmitted to the muscles. When the nerve signal reaches the nerve terminals, you have release of a transmitter substance called acetylcholine. Acetylcholine binds to receptors on the muscle side, and that causes positively charged ions to flow into the muscle, which eventually leads to muscle contraction. We also have other ion channels residing in the muscle membrane that are very important for the transmission of signals from the nerve to the muscle. And one of these ion channels is called CLC1, which is a chloride channel. The chloride channel's role is to inhibit transmission. So it functions as a brake. And the goal with NMD670 or any CLC1 inhibitor for that matter is to loosen the foot on the brake, allowing signals to come through when there is an impaired ability to transmit signals from the nerve to the muscle. We have already shown that this treatment can be leveraged in myasthenia gravis, which is another neuromuscular disease. In this study, which was recently published, we showed that one tablet could improve muscle strength and muscle function in the hours after taking the dose. In parallel with the study in myasthenia, we also conducted observational studies in Charcot-Marie-Tooth disease. We conducted preclinical investigations that have paved the way for the clinical development of NMD670 in Charcot-Marie-Tooth disease. The study is currently being put together in a manuscript. It will be submitted very shortly, but the studies and data are not publicly available yet. Nonetheless, I want to take the opportunity to share some of this very new data with you, because it's important to understand why we are investigating NMD670 for the treatment of Charcot-Marie-Tooth disease. So the first part of this study, the translational study, investigated whether participants and adults with CMT have impaired nerve-muscle communication. And we used a highly specialized EMG technique called a single-fiber EMG that allows you to ascertain that very particular aspect is the transmission of signals from the nerve to the muscle impaired. And what we saw was a very pronounced impairment in the ability to transmit signals from the nerve to the muscle, regardless of the type of CMT, essentially. So, as you see in the figure here, the higher up you are on the graph, on the y-axis, the more impairment you have in the ability to transmit signals. We went one step further and looked at the association between the impairment in the ability to transmit signals and the clinical symptoms. And we saw quite striking associations between impaired nerve-muscle communication and clinical symptoms such as muscle weakness, muscle fatigue, balance, and mobility. We have also done preclinical work. I'm here sharing data from one preclinical experiment which was conducted in a CMT2D mice model of CMT. And here we showed that when we gave NMD670, we could increase muscle strength in these animals that were affected with CMT2D. We did comparable studies in a mice model of CMT1A also. And so really these data very much supported the clinical development of CLC1 inhibitors for the treatment of CMT. And based on those exciting results, it was decided by the company to progress NMD670 into clinical development for the treatment of CMT. This here is showing you the very high-level study design of a phase 2A proof of concept study that is just about to start with NMD670 for the treatment of CMT. The goal of the study is to assess the efficacy and safety and tolerability of NMD670 given twice daily over a period of 21 days. There will be a screening period of up to 28 days, and then eligible participants will be randomized to receive either a placebo given twice daily or NMD670 400 milligram given twice daily. It is the goal to enrich the study with at least 30% of the participants that have CMT type 2, because we want to ascertain whether this population of CMT participants respond equally well or maybe even better than their CMT1 counterparts. The primary endpoint of the study will be the 6-minute walk test and the total distance that can be walked over 6 minutes. We include a range of other efficacy assessments, and many of these assessments are only possible to include because of the natural history studies that were conducted in this space, in particular the ACT-CMT studies. So this here is showing the list of US trial sites that will participate in the study. We have a total of 19 sites across the US, Belgium, France, Denmark, and Spain. And we have at least 8 sites that we hope to include in the study in the US. We already have 2 trial sites that are activated for enrollment. These are the trial site in Austin with Dr. Hussain and the trial site in Columbia, Missouri with Dr. Arnold. For more information about the trial, including information about the trial sites, contact information, you can go to clinicaltrials.gov. You can scan the QR code that will take you directly into the trial information webpage, or you can also write down the NCT number, which is also written on the slide. You're also more than welcome to contact me directly, and then I'm happy to answer any questions or direct you to a trial site if you're interested in participating in the study. And with that, I would just like to extend a sincere gratitude to all the families and patients that participated in the studies that we already completed, to all of you for listening, to the investigators and the site staff that are performing the studies that we are running now, but also those that we completed, and of course to NMD Pharma and not least to CMTA for this fantastic opportunity to address you all. So thank you very much.
Thank you, Thomas. We're using this one, yeah, okay. Right, so now we have about 21 minutes for Q&A. So whatever questions you all have for us, we'll be happy to try to answer them.
Well, first thing I have to say is really not a question so much as a thank you. I know y'all have come from all over. I mean, Denmark, Baltimore, and Miami. So thank you for taking your time. And I know that all the work you put into this is, please know that it is appreciated. So thanks. I have so many questions. Let's see if I can remember any of them. So the first one is, in terms of the drug you were just talking about, does that have applications. It sounded like for CMT subtype 1 and 2, and if someone was interested, is going to that QR code. Is that a way to sign up?
Yeah, I can answer that, sure. So it is indeed correct that we think the drug has applicability across CMT types. In this initial study, the requirement is to have a genetic confirmation of disease. You need to have CMT type 1 or CMT type 2. And if you have, you are potentially eligible for the study. In order to be enrolled, we need to put you in contact with one of the trial sites. Once sites are activated, it will be listed on clinicaltrials.gov. There will be information about study coordinators that you can contact directly. Until then, you're more than welcome to contact us for further information.
Great, thank you. And then the other quick question is, it really relates to me trying to explain my story and how CMT works, but also, I guess, just being able to understand it at all. So I've always told people, for some reason that the nervous impulses, drop off, and we really don't know why that is. And I hear you mention chloride channels and things like that. I do have a background in biology, but still these things are extremely complicated. So I'm really hoping for maybe a simplified narrative of why does CMT work the way that it does, specifically in type 2, where we know that it's not due to the loss of myelin that we are seeing decreased function. So I don't know if you guys have a simplified, kind of explanation of that, and also maybe why do we see a drop off in functionality where we do when we do?
Sure, no, that's a great question, and it was missing on my thought. But so for type 2 CMT, the issue is the number of axons that are there to transmit information, right? So we know there's a correlation between how many axons you have, how many muscle fibers are activated, and how much strength you're able to generate, or how much sensation you're able to convey back. So we know that as the nerve damage process progresses in type 2, the number of axons that you have in your nerves drop down because they're not able to function anymore. Some of them will be there, but just not functioning. Some of them will be lost because they can't sustain themselves anymore. And then with time, that drop out of axons will start to translate into less strength generated by your muscles, because that strength needs the order, the command coming from your brain through your axons to happen. So in a nutshell, type 2 CMT is an issue of a loss of your axons in your nerve fibers. Does that make sense?
Yeah, absolutely. I think my question again could have been the curious type is, what is causing the loss of axons?
Well, so that depends on the type of CMT you have. And that's kind of really is what this kind of scientific community is working on, because there's very likely different mechanisms for different genes. And that's what makes this whole field very, very challenging, because in the case that Thomas presented, there may be some areas of commonalities, but in most cases, especially if you go very close to where the problem is originating from, that will be very different from type to type. So if you have type 2A, you have an issue with your mitochondria, how that kind of part of your cell that generates energy works and kind of traffics towards your nervous system. If you have 2E, which is a mutation in a neurofilament, which is a protein that helps kind of maintain your axon integrity, you have issues with that. So there are very few kind of overarching areas that we could put into a single, okay, this is what's going on. It really depends on the type that you have. I don't know if there's anything to add, but just another question there, and there, and there.
So just kind of following up on what you said with the axons kind of dropping off, let's say, in terms of their ability to communicate or really just function. Let's just take as a starting point, Thomas, your initiative, right, and your company's initiative to sort of have these inhibitors that hopefully will restore or at least maintain function. I guess that's really my question. In the case of something like that with these inhibitors, is it a matter of then, let's say it works, it preserves the axons that work, or can you bring the axons that are not right now in play back into play?
Let's let Thomas answer first, and then we can get some context.
Yeah, no, sure. Thank you a lot for that question. So this drug will most likely not address the underlying cause of the disease. It will not address the axonal loss directly. This drug will, via improved nerve-to-muscle communication, increase the ability to contract your muscles acutely. It may be that there are additional effects. We are studying whether it could also have re-innovating capabilities, but it's very early days for that. It's something that we are looking into.
Very clear.
And I'll add to that as well. So the way I think of this NMD 670 drug is, I think of it as something that could potentially address a complication of CMT that is seen across CMT subtypes. And if this medication, really does successfully treat some of these CMT symptoms, I see it as a medication that could potentially be used in adjunct with other medications that are more targeted to the underlying abnormality in specific CMT subtypes.
And just finishing up, so that I kind of use that to help understand this concept. So there are different ways that you can treat a genetic condition. You can focus on where the problem is really originating from, in the DNA, the mutation, the protein, but you can also focus on the long-term consequences. And I think this treatment is really focusing on that kind of right side of the slide. It's not going to cure the problem itself, but it's very likely that even if we manage to cure the genetic problem, depending on where the patient is in the kind of progression of their disease, we have to definitely tackle the long-term consequences as well. So to be Pasha's point, we're probably going to need this combination of treatments where we correct where the genetic problem is, and we're becoming better and better, having tools to do that, but also starting to focus on how can we make axons regrow, neuromuscular junctions function better, myelination to happen again. So those are things that we're always thinking about, because it's probably not going to be one single solution, especially if you're not able to treat before patients develop symptoms.
We have a question over here. Hi, thank you. My son has CMT type 2, and supposedly the underlying cause is mitochondrial disease. And his progression has been rapid in this last year or two. And I was wondering, if he got into this study, could it slow down that progression at least?
So I'll just start, and then I can give you some. So that's kind of one of the things that we need to answer with our trials, is what can realistically be accomplished, especially depending on where each patient is in terms of their disease progression and symptom. For the treatments that are focusing on treating the genetic problem or the kind of biological problem that's caused by the mutation, our expectation is to at least stabilize the disease, right? We can't be sure, because we're only going to be able to tell you if that's happening once we have results of a trial. But the peripheral nervous system has some plasticity, meaning it's able to regenerate to some degree. That ability varies depending on the patient's age, how healthy they are, and how severe the damage is. So that's to say that there's a potential that once we are able to stabilize a disease, that some recovery can happen. But that's all very theoretical at this point, because we haven't had the opportunity to see a CMT type stabilize and then see what happens over time. So it's an open question, unfortunately, yet, but I think we're going to be able to start answering those once the trials that are ongoing and are about to start will kind of provide us some answers.
So for him to get into a trial, how we get the steps here somehow as to how to get into this trial.
So in the case of this particular trial, this is the first proof of concept study. It will only be enrolling adults. If it's successful, we will be rolling this out to other types of the disease, to pediatric populations and adolescents. But for this initial study, we are only enrolling adults.
Hi, are there any updates on CRISPR or gene therapy that you can share for type 2?
So there's a lot of preclinical work ongoing. So there are different groups. Our group works on a specific approach using antisense oligonucleotides for type 2E. There is a work at Gladstone Institute in San Francisco working on 2A and 2E. So there's a lot of work ongoing, but still on the preclinical stage. We don't have anything that have reached a point that we are certain of. One, the safety, which is the most important thing that we have to determine before even starting to consider in human testing, but also whether this approach would work for these different types of CMT. So there's a lot of work, and there's a lot of groups working across the globe on different genetic approaches, either gene replacement therapy or CRISPR and other types of kind of genetic modifications, but we're not there yet. The only exception is for CMT2S, which is a very, very particular type of CMT where the company is working on a gene therapy, and I think they're reaching clinical testing.
Can an individual have two subtypes of type 2, or could two siblings have two different subtypes of type 2?
So the short answer is yes, it's possible, because one type doesn't protect you from having another type. It's just exceedingly rare. So it's more common to see patients have one very clear type of CMT and then carry what we call variants of uncertain significance in other genes. And those variants do not mean that the patient has those other types of CMT, just that they carry variations in their DNA that's unlikely to be necessarily disease-causing, but they are there. So the short answer is yes, it's possible. We have seen families where we see two types of CMT kind of coming from different branches and then meeting up in certain family members, but it's very, very, very rare.
Hi, I have 2E, as does many members of my family. I obviously, as an adult, would like to sign up for a clinical trial. How do I go about doing that? I think you may have mentioned it, but I missed that.
Yeah, sure. So for 2E, the work is still preclinical, as Dr. Saporta was saying. So there isn't a specific clinical trial for a disease-modifying therapy for 2E at this moment. There may be in the future. But, you could potentially be a candidate for this NMD 670, NMD Pharma 670 compound, which would be applicable to people with types 1 and type 2. And so let me hand it off to Thomas to talk more about how to sign up.
Sure. Thanks for the question. So the way to go about this would be to get in contact with one of the study coordinators at one of the sites that are enrolling for this study. And then they will contact you for a screening visit. At that screening visit, they will determine if you are eligible for the study or not. And then if you are eligible, you would be randomized into the study. We are more than happy to help you get in contact with one of these sites.
I have a question about that. So the sites were noted in like the third to last slide. I happen to know one of the locations in Spokane, Washington. I know I've worked with that doctor quite extensively. So should I contact him and just say, here's what I've heard, and I'd like to be considered for trial?
Yeah, you're more than welcome to.
Okay.
Just to finish up on this, because think they're very important, just general ways to go about this. So the first thing is knowing that something's happening, right? So being connected with the CMTA, with just kind of the social media environment for CMT will help you at least know that opportunities are there. And then you can see if that opportunity applies to you, right? So if there's a clinical trial for type 1, you know it's not for you. But if there's any opportunity for type 2, then you know, okay, at least you know that this is going on. And then there are different places where you can find very detailed information of the trial. There's a website called clinicaltrials.gov that will list every trial that's ongoing in the States or that has FDA kind of oversight. And you can go to that website, put CMT or Charcot-Marie-Tooth, and you'll see a list of what studies are ongoing in the US, in Europe, in other places. And that will allow you to understand, okay, what is the study about, what they're testing, who are eligible, because it will vary. Some will be studies for pediatric patients, other for only adult patients, for patients with that degree of weakness or severity. So understanding eligibility criteria is important as well. But always work with your doctor, because especially if you're in a center that has some kind of CMT, either center of excellence or at least some CMT expertise, but even they don't, your physician should be able to help you figure out, is this trial right for me, like the commercials will say, but it will allow you to know what opportunities are out there and who should you contact to go about being considered for that.
I think we have time for maybe two more. How about you? And I'll get over to you.
So I have CMT2F and I also have MS. And I know that I was researching and they think that there might be a connection between the two with the HSP27 heat shock protein. Is there any more research going into that? Because of even trying to get help, I struggle with people. Was this the MS or is this the CMT variant and not getting help from anybody? Because which neurologist do I go to?
I'm not aware of a lot of research on that particular combination. I don't know if I should know. So it's a very, very particular situation. So it's tough. And I can completely feel your pain in that sense because it is difficult. Do you have like a CMT specialist and an MS specialist? Okay, so I think they will be your resources to try to navigate. This is a very complicated situation because there are symptoms that are very overlapping. There are some ways that you can go about trying to figure out, but you're definitely going to need the help from very experienced and expert people to navigate that. There's no easy answer for that. I'm sorry, but and I'm not aware of specific research to try to really tease out what the connection is. But from a therapy perspective, of course, the MS is way more treatable than your CMT. So I guess the focus for now will be making sure that your MS is under control.
Thank you.
Thank you again for everything you do. So I have a two-part question. The first question is, with what you've explained with the axonal CMT, it generally begins distal, hands, feet. Our 16-year-old son has a report that confirms CMT SORD. However, he's having issues with his hip. Is there something that signals that it could affect the hip region due to CMT, or that might be something else? So that's part one question. And the second part of the question, he's very active. He plays basketball, very, very, very good in terms of his muscle. However, since you're doing this clinical trial only for adults, how would I be able to stay connected with you once he turns 18, or if you're doing pediatric? What's the best way to stay in touch for the future?
So just answering the second question first, because we kind of touched on this a little bit. So the first thing is making sure that you have a neurologist or a specialist that is managing him, his CMT, and that should be your kind of gate towards any opportunity for research. But the same thing applies where being active on social media, being connected with patient foundations, just kind of knowing what's out there would help. Because sometimes, we see hundreds of patients a year. We try to keep track of what's going on for every single, but I'm surprised half of my day for patients coming and saying, oh, doctor, there's this study here. What do you think about that? So they will bring studies to our attention sometimes. So it's important to have this to two-way kind of communication. But that's kind of what I would recommend doing. The other question, so hip issues with CMT, there are two different ways that this could happen. Just during development, we know CMT is a developmental problem, right? So it's going on for very early on, and depending on the CMT type, it's even more true than for others. So your hip can form differently if you have CMT, and that's what hip dysplasia is. So that's one possibility, and it's common in CMT1A and other types of CMTs. But even more common than that is if a patient have issues with either foot drop or weakness in their ankles, they will try to compensate, right? So they're going to start using a lot of their upper body or at least upper leg to be able to walk without tripping and falling. And that will overload the hip joint, the lower back. So the first thing I do once we rule out that the patient has actually a joint problem related to the CMT with an X-ray, for instance, is making sure that they are properly braced or they are not having issues with a foot drop, even a mild foot drop that may be causing this kind of adaptation to happen, which is not going to work well. Correct. Yeah. All right. So I think we should be done. Yep. Thank you so much for being here.